This recording is the Anatomy of the Nervous System, Part 2 of 3. The spinal cord extends from the medulla oblongata just at the foramen magnum of the occiput to approximately the level of L1 to L2 in the lumbar vertebrae. The cord, as a cord, ends at that level in a cone-shaped structure that is often referred to as the conus medullaris. You can see that term here on the left. From there, the cord splits into fibers, forming a structure referred to as the cauda equina. There is a central portion of the conus medullaris that continues and is covered by the innermost layer of meninges known as the pia mater. And this continuation of the conus covered by pia mater forms a structure known as the filum terminale, which extends all the way down to the coccyx and attaches to the coccyx, anchoring the spinal cord all the way to the coccyx. You can see the term filum terminale also on the left of your screen, right here. You'll notice by looking at this picture that the cord itself enlarges in the cervical region, forming the cervical enlargement, as well as in the lumbar region, forming the lumbar enlargement. If you stop and think about the fact that we have to have more nerves going to our arms and our legs for motor control, it makes sense that there would be enlargements in those areas of the spinal cord. The spinal cord runs through the vertebral foramina of the spinal column and is approximately 18 inches in length. It's protected by bone, meninges, and cerebrospinal fluid, as well as by fat, which helps protect it within the vertebral foramina. There are 31 pairs of spinal nerves that come off of the cord through the intervertebral foramina, which are located laterally in the vertebral column and open up at a 45-degree angle in the cervical vertebrae. A mixed nerve is a nerve that carries both sensory and motor information. We already mentioned that the spinal cord ends as the conus medullaris around the level of L1 to L2, at which point it becomes the cauda equina, which means horse's tail. You can really see the horse's tail or cauda equina in this picture. The filum terminale is an extension of the conus, which we see in the very center, and is covered by the innermost layer of the meninges. There are 3 layers of meninges, which are protective tissue coverings that surround the brain and the spinal cord. And the innermost pia mater covers the filum terminale, which, as I mentioned previously, attaches and anchors the spinal cord to the coccyx. We already discussed the cervical and lumbar enlargements, and we also discussed that there are 31 pairs of mixed spinal nerves, which means they carry both sensory and motor information coming off of the spinal cord laterally to other parts of the body. Each spinal nerve has roots forming the nerve. There is one from the posterior or dorsal aspect of the cord called the dorsal nerve root, which carries incoming sensory or afferent with an A information, and there is a ventral nerve root on the anterior side of the spinal cord that is going to carry descending efferent with an E motor information. In the top picture on the upper right-hand side, you can see the ventral root, which technically forms from smaller rootlets coming off of the spinal cord that converge to form the ventral root. There are also dorsal rootlets which converge to form the dorsal root. You can see the term dorsal root on the left-hand side at the top. If you noticed, you'll also see the term ganglion, because at this area of the dorsal root, we find an enlarged structure called the dorsal root ganglion. In the central nervous system, we call a collection of soma, or neuron cell bodies, either a nucleus or a ganglion. In the peripheral nervous system, we only use the term ganglion when we're talking about a collection of soma, neuron cell bodies. Because the posterior or dorsal root is carrying incoming sensory information, these soma are coming from the unipolar or pseudo-unipolar sensory neurons. I'll attempt to draw one to refresh your memory. Here are the dendrites of that pseudo-unipolar neuron. We have a fiber, the soma, and then it continues in this direction to eventually synapse with another neuron. When it comes to incoming sensory information, usually there are 3 neurons involved in receiving and getting information up to the primary somatosensory cortex. So we talk about first order, second order, and third order neurons just for sensation. Here we have the soma or cell body of that sensory neuron, and in this picture on the bottom left, these little circles represent the soma of those unipolar sensory neurons. That's why the structure is enlarged, because the soma tend to be circular or oval in shape. That is a dorsal root ganglion. It's got the cell bodies or soma of somatosensory neurons. The dorsal root and ventral root will merge together. Here we have the dorsal root and the ventral root merging together to form a spinal nerve. Remember, the spinal nerve is going to have information that's both sensory and motor, therefore we say it is mixed. So sensory information is afferent or afferent and is ascending because we've got to get it up to the brain to figure out what we're sensing. Motor information, which is coming out from neurons that actually have their soma in the anterior horn of gray matter, here in the lower picture in green. That's where the soma of motor neurons are going to reside, and their axons will go out the ventral root. So motor information is efferent and descending. Remember, sneak sensory in the back door, motor out the front to catch the bus. On these pictures, we'll also notice some structures and define and demonstrate them. In the anterior portion of the spinal cord, we can see that there is a crack or fissure. This is the anterior median fissure. The term fissure simply means a crack. On the posterior aspect, we have a groove that runs the length of the cord called the posterior median sulcus. If we look at the cord, you'll notice that the white matter is on the outside and the gray matter is on the inside. This is the opposite of what we saw with the cerebrum and the cerebellum, where the cortex was gray matter on the outside and the inside had white matter, with the exception of a little bit of gray matter that was buried internally. The gray matter consists or exists in structures that are known as horns. So we have a posterior gray horn. The posterior gray horns, by the way, tend to be a little bit more pointy in nature and tend to go to the perimeter of the cord. We have an anterior gray horn. The anterior gray horns tend to be a little bit more rounded. And in some areas of the spinal cord, we'll find a lateral gray horn. Here you can see that laterally. So we've got white matter on the outside, gray matter on the inside. You'll notice that there is a central canal in the middle of the gray matter. And that central canal is filled with cerebrospinal fluid. It runs the length of the cord. The central canal is in the center of a structure that joins both sides of gray matter called the gray commissure. This area right here surrounding the central canal connecting both sides of gray matter is referred to as the gray commissure. Here we've got a picture of the spinal cord under the microscope. I can tell just by looking at this picture that this is the posterior horn of gray matter because it appears pointier. The anterior horns of gray matter appear more rounded. So this would be the anterior horn. We have the central canal as that white dot in the center, surrounded by the gray commissure on either side. Because the anterior horn is here and the posterior horn is here, that would make this the anterior median fissure, and that would make this the posterior median sulcus. So when we look at the posterior horns of gray matter, or dorsal horns of gray matter, these areas of the spinal cord basically contain interneurons of somatosensory and viscerosensory neurons. Remember, an interneuron is just a neuron between other neurons. And with sensation, there are usually 3 neurons involved in getting sensation from a part of the body to the brain where it can be processed. So we have a first, second, and third order neuron. The ventral gray horns or anterior gray horns are where the cell bodies or soma of motor neurons are going to reside, their axons exiting out through the ventral root to go to a skeletal muscle. So technically, these guys are lower motor neurons. With contraction of skeletal muscle, there's 2 neurons involved. There's one that comes from the primary motor cortex, which is the upper motor neuron, and then the lower motor neuron is the one that you see here in the anterior horn of gray matter, because that's going to go right to the skeletal muscle. We know that gray matter is going to be larger in the cervical and lumbar regions, again because of the arms and legs, and there are also lateral gray horns that are more developed in some areas of the spinal cord, which will contain the soma or neuron cell bodies of autonomic motor neurons. So here we have skeletal muscle motor neurons. In the lateral horns of gray matter, we're going to have the cell bodies of motor neurons that go to the organs. So that's autonomic. They're still going to go out through the ventral route because we always motor out the front door. However, autonomic motor means motion for organs that you don't normally control voluntarily. This will take us to the white matter in the spinal cord surrounding the gray matter. In this particular picture, the white matter is colored either blue or red to determine that in the case of the blue encoded areas of white matter, we're looking specifically at incoming or ascending sensory information. Ascending is afferent with an A, and it's always going upward, whereas the red descending tracts are for motor information. And you'll notice that the dorsal white columns, which are found in the posterior aspect of the spinal cord, include a lot of area of the spinal cord. And the dorsal white columns are basically carrying incoming sensory information about body position, temperature, pain, touch, pressure, vibration. Also, visceral sensory information is coming through these tracts. Remember, a tract is a bundle of myelinated axons going in the same physical direction. So they're, in this case, all ascending. And the terms fasciculus gracilis and fasciculus cuneatus, you don't need to know. I'll just explain it quickly for you so you do understand it, though. The fasciculus cuneatus is basically taking incoming sensory information from the lower part of your body and your trunk up to about the level of T6. And then the fasciculus gracilis adds to that from the upper portion of the trunk and limbs and head and neck. It gets added in from the fasciculus gracilis. Now, you'll notice that there are different terms on this picture, and I want to point this out to you because it's really important that you understand this. You can easily tell by the word whether it's sensory or motor. If it's going up, it's sensory. If it's going down, it's motor. So ascending is afferent, sensory. Descending is down, it's efferent, motor. How do you tell? Look at the words. The words will tell you everything, even if you don't understand all the parts of them. Let me show you. If we look at the word tectospinal, well, the first part is tecto. Even if you don't understand what that is, it ends in the spinal cord. So it's got to be coming from above, going down, ending in the spinal cord. That's descending, it's motor. Okay, and we can check ourselves. Look, it's red, it's descending. Let's look at this term over here that says spinothalamic. Well, even if I didn't remember the thalamus in the brain, if it starts in the spinal cord, it can only go up. Because where it comes to these terms, they're either somewhere in the brain, or the cerebellum, or the spinal cord. So if it starts in the spinal cord, like spinothalamic, it's going up. If it ends with spinal, that means it's coming down. So spinothalamic, ascending sensory information. By the way, the lateral spinothalamic tract is super important because that actually helps us understand life-saving information about pain. So if we're faced with something that's really painful and damaging, really damaging to tissue, the lateral spinothalamic tract is the one that's going to carry that information. That's important for you to know. Lateral spinothalamic tract, life-saving information about pain so you can get away from it. Okay, let's look at another term. Let's look at spinocerebellar. Okay, from the spinal cord to the cerebellum, still going up. How about rubrospinal? Well, if you didn't know, rubro meant the red nucleus. That's fine, okay? In the brainstem to the spinal cord, doesn't matter. You know that it ends in spinal, so it's ending down. It had to come from up to get to down. So descending motor. That's how you figure these things out. You can tell by the words. Okay? So again, the columns of white matter are divided and named funiculi. Each funiculus, which is singular for funiculi, has several tracts with similar destinations and functions. Remember, sensory is going to require 3 neurons to get up here. Motor, 2. You have an upper motor neuron and a lower motor neuron for motor control for voluntary skeletal muscle. This takes us to the blood-brain barrier. The blood-brain barrier is important because we know that the chemicals in blood are toxic to neurons. So the blood-brain barrier helps prevent the stimulation of neurons that could occur accidentally if stuff could leak out of the capillaries. Remember, capillaries are teeny tiny blood vessels that are taking oxygen and nutrients to tissues of the body. However, we don't want everything to just go right through the capillaries when it comes to nervous tissue. It could kill the nervous tissue. We don't want that. That would be bad. So the blood-brain barrier includes the endothelial cells, which are the cells that make up the walls of the capillaries, being joined together or connected by tight junctions. Tight junctions keep things from leaking through. In addition to the tight junctions that hold the walls of the capillaries together, we also have the podocytes, P-O-D-O-C-Y-T-E-S, podocytes of astrocytes, which are those star-shaped cells, which also help to wrap around the capillaries. So that helps keep things in the capillaries. Now, the astrocytes can release permeability controlling chemicals, which may allow certain ions to flow into the astrocytes, so the astrocytes can transport them through to the neurons. That's transcytosis. And only lipid-soluble compounds can get through any membrane, because remember, membranes are lipid-soluble. So if we have things like oxygen and carbon dioxide, well, they can go through membranes without any assistance because they are fat-soluble. They're lipid-soluble, goes right through. Glucose, lipid-soluble chemicals. Lots of hormones are lipid-soluble. Alcohol is lipid-soluble. And so these types of things can also go through easily from the bloodstream to the neurons. Large water-soluble molecules cannot cross the blood-brain barriers, so things like red blood cells, white blood cells, proteins can't cross. And this is a way that helps us control the transport of ions in order that neurons can fire appropriately. Interesting that dopamine as a neurotransmitter cannot pass through the blood-brain barrier. It has to be released at a synapse. This takes us to the blood supply of the brain, and there's lots of different ways for blood to get to the brain, which is important. We know that neurons are highly metabolically active, so they need lots of sugar for adenosine triphosphate to be produced in the mitochondria through the process of cellular respiration, and they're going to need lots of oxygen for this process as well. So we've got to have multiple ways to get blood to the central nervous system. We've got to have a continuous supply. And there are 2 sets of arteries that supply the brain. We have arteries that are called the internal carotid arteries, as well as the vertebral arteries. And in case you don't remember, the cervical vertebrae are the only vertebrae that have transverse foramina. Those are holes in the transverse processes that allow for a passageway for the vertebral arteries. That's how they got their name, because they actually go through structures in the vertebrae. So the internal carotids and vertebral arteries are going to help supply the brain with blood, and these arteries converge into a circular structure known as the circle of Willis, which is also called the cerebroarterial circle, that helps to maintain blood flow or perfusion to the brain, even if one of the parts of the arteries gets blocked or narrowed. Once oxygen has gone through the brain and nutrients have been taken from the bloodstream to the neurons, then we need a way to get the venous blood back away from the brain. And after circulation of blood goes through the brain, the venous blood will start to collect in a series of structures known as sinuses. And we've got different sinuses. We've got the superior sagittal sinus. We have an inferior sagittal sinus. And we have a straight sinus. And we have an occipital sinus. And these sinuses are all going to drain into a structure known as the confluence of sinuses. Think of the word confluence. Con means with, fluence, with fluidity, okay? So all this flow from the superior and inferior sagittal sinuses, the straight and occipital sinuses, is going to merge into the confluence of sinuses. And then from there, venous blood will flow into the transverse sinus and then into the sigmoid sinus, where it will enter into the internal jugular veins going back down towards the heart. Kids, your father and I have discussed this at length, and we feel it's time we tell you about meninges. The meninges are the protective tissue coverings surrounding the brain and the spinal cord. They're made up of dense irregular connective tissue, and the outermost layer of the meninges is called the dura mater, or tough mother. The dura mater exists in a single layer around the spinal cord. However, in certain areas of the cranium, it actually splits into a periosteal layer, which is next to the cranial bone, and a meningeal layer, which is close to the next deeper layer of meninges. And the dura mater helps to, again, simply protect and enclose the entire central nervous system as well as its major blood vessels. The next layer of meninges that we find deep to the dura mater is the arachnoid mater. Arachnoid means pertaining to spiders. The arachnoid mater has this spider web-like appearance because it has these trabeculae, or lace-like fibers, lattice-like fibers, that project between it and the innermost layer of the meninges. And deep to the arachnoid mater, we find the subarachnoid space. Remember, the prefix sub means under or below. It is in the subarachnoid space that we find cerebrospinal fluid circulating around the brain and the spinal cord. So cerebrospinal fluid circulates in the subarachnoid space. You'll notice that in this picture on the right-hand side, there's a term that says arachnoid granulation villi. It's pointing to one. Villi is actually plural, so technically it should be arachnoid granulation villus. And what happens is there are portions of the arachnoid mater and their trabeculae that actually extend up into the superior sagittal sinus. And this is important because as cerebrospinal fluid is being produced continuously, in order for the prevention of too much pressure to build up, as the fluid is produced, some of the fluid will be pushed out through the arachnoid granulations into the venous blood of the superior sagittal sinus. What happens is we actually produce cerebrospinal fluid from tiny little blood vessels and the fluid in them. So the fluid for cerebrospinal fluid comes from the bloodstream, and then it goes back into the bloodstream. The innermost layer of Meninges is the pia mater, which means tender mother or gentle mother. And the Meninges extend into every convolution of the central nervous system. We know that the conus medullaris is the end cone-like structure of the spinal cord, and there's an extension of that conus medullaris that extends downward and is covered by the pia mater. The pia mater also has little tooth-like projections outward laterally to attach it to the dura mater. Those are called denticulate ligaments. These structures help to anchor everything to the bony structure and to the dura mater. Now, as we look at the meninges, they actually extend inward and across certain structures where there are places for it to fold inward to provide more protection. And we know that if we were to go down the longitudinal fissure between the cerebral hemispheres, we could go down as far as the corpus callosum. And so there's actually an extension of the meninges that goes between the cerebral hemispheres to the point of the corpus callosum, and that extension of the meninges is called the falx cerebri. It helps to separate the two cerebral hemispheres to a certain point. There's also a falx cerebelli, which goes between the two hemispheres of the cerebellum. And then there's another extension of meninges that forms a tent-like structure between the cerebrum and the cerebellum, and that is the tentorium cerebelli, which separates the cerebrum from the cerebellum that you can see on this picture on the left. We previously mentioned that cerebrospinal fluid circulates in the subarachnoid space, which is between the dura mater and the arachnoid mater. You can see that in this picture flowing here. The purpose of cerebrospinal fluid is to cushion and support and transport nutrients and waste in the central nervous system, and it's derived from blood plasma. It just has fewer proteins. It's monitored by the brain for changes and is found in fluid spaces such as the subarachnoid space, as well as the ventricles and the central canal of the spinal cord. If we look at this picture, it's a little bit tough to see, but you can kind of make out the ventricles, which are the fluid-filled spaces in the brain. They're kind of in a brownish color in this picture. Let's see if I can turn them green. This would be the lateral ventricle. And if you've seen this in lab, you know the lateral ventricles have a kind of a horseshoe shape to them. They're joined together to each other in the third ventricle by a structure called the interventricular foramen. So that joins them to the third ventricle. The third ventricle has a canal called the cerebral aqueduct, which joins to the fourth ventricle. And there are openings that allow cerebrospinal fluid to go from the fourth ventricle to the subarachnoid space, as well as down through the central canal of the spinal cord. So cerebrospinal fluid is basically going to be produced and circulated by the ependymal cells that are found just next to the choroid plexuses. So the term plexus means network, and choroid means resembling red. Let's take a closer look at this on this picture before going to the next slide. If I erase some of these highlights, you can see some of the network of choroid plexus in magenta here. Associated with the fourth ventricle and here on the underside of the lateral ventricles. You can really see the network of choroid plexus on the mold of the ventricles if you look in lab, depending on which lab you attend on one of the campuses. I know the Marietta lab has a really nice example of the ventricles, and you can see the choroid plexus associated with those ventricles. Here we have a close-up picture of part of the choroid plexus. Remember, it's a red network because it's made up of little capillaries, which are blood vessels. And as blood flows through the capillaries, some of the pressure of the blood flowing through these capillaries is going to force a little bit of the fluid portion, the plasma portion is the fluid portion, to leak through, and it will be absorbed by the ependymal cells. And so as it filters through the wall of the blood vessel, that filtrate entering into the ependymal cells is going to contain things like glucose and oxygen and vitamins and ions like sodium chloride and magnesium. And then the ependymal cells will absorb the filtrate and further modify it to make sure that the cerebrospinal fluid has the correct components before it starts circulating. So these ependymal cells are actually on the surface of the choroid plexus between the plexus and the actual inner portion of the ventricles. Sometimes it's important to test the cerebrospinal fluid if there's a problem like bacterial meningitis being suspected, where we might see infection in the cerebrospinal fluid as the meninges may become infected. And so the test for cerebrospinal fluid is referred to as a lumbar tap or puncture. You can see the needle involved in this thing. And usually it's done in the subarachnoid space below L1 and L2 because that's where they have the best area to go into the subarachnoid space without damaging any of the tissue. So signs of problems could include things like red blood cells, white blood cells, and proteins being found in the cerebrospinal fluid, because remember, those types of molecules don't normally exist. So the presence of those types of molecules could mean anything from an infection or a tumor or a hemorrhage. The circulation of the cerebrospinal fluid, remember, it's going to be formed from blood plasma. It starts as fluid gets leaked through the capillary walls that make the choroid plexus, and then the ependymal cells soak up that fluid and make sure that the ionic concentration and fluid is appropriate for cerebrospinal fluid. And so it goes into the lateral ventricles through the interventricular foramina to the third ventricle, then to the cerebral aqueduct to the fourth ventricle, into the central canal of the spinal cord and subarachnoid space. As the fluid keeps getting produced, because blood flows 24 hours a day, 7 days a week, so cerebrospinal fluid is continuously produced. Remember those little projections called arachnoid granulation villi that poke up into the superior sagittal sinus? So some of that fluid will be pushed out of the subarachnoid space at the arachnoid villi into the venous blood in the superior sagittal sinus. So that's how that gets produced and flowed and gets circulated back into the cardiovascular system. It comes from blood, goes back to blood. This takes us to one of the problems that can occur with the flow of cerebrospinal fluid. The term stenosis means a hardening or narrowing of, and so aqueductal stenosis, remember the cerebral aqueduct, which takes fluid from the third ventricle to the fourth ventricle, and then the fluid actually exits the fourth ventricle to go to the subarachnoid space or down the central canal. So if the cerebral aqueduct gets stenosed or narrowed, that can significantly decrease the flow of cerebrospinal fluid, causing a condition of hydrocephalus or water on the brain. And it's often treated surgically by inserting a shunt system to divert the flow of cerebrospinal fluid from the central nervous system to another area of the body where it will simply be absorbed. Traumatic brain injuries include things like concussions, where a brain injury is usually caused by a blow to the head, and multiple concussions can cause cumulative damage that occurs exponentially as opposed to linearly. So it's really important for anybody who has a concussion to heal more completely than was originally thought years ago, because they found that repeated concussions can cause a lot more damage to the brain than previously understood. Strokes, or cerebrovascular accidents, that's what CVA stands for, cerebrovascular accidents, are either caused by a lack of blood to the brain tissue from ischemia, which a blood clot can cause the blood to stop flowing, and that can result in death of the neurons in the brain. Or a hemorrhagic stroke can also cause the nervous tissue to die. We can see in the picture on the right an example of a hemorrhagic stroke. So there are two different kinds of strokes. They can do damage either way, either no blood, where there's no oxygen and nutrients getting to the neurons, or bleeding, where the blood alone can damage the neurons. And the picture on the far left on the bottom can show you the difference in someone in terms of paralysis that can occur with a stroke or cerebrovascular accident. Usually what's noted is hemiparalysis, where half of the body is paralyzed because most of the motor information is coming from the opposite side of the brain. So if my right side of my body is affected, then my left side will experience hemiparalysis. Now remember, 80% of the neuron for motor control goes to the opposite side, but that also means that if a person works on it, he or she can regain motor control by retraining the same side of the body to be used for that motor control. And that's it for this slide presentation. We're going to go on to part 3 of 3.